this body? What is it?
Fleming of St Mary's Hospital, London, who discovered that it produces the drug known as penicillin.
The question was, which drug would go with which cell?
It's a collaboration that's unexpected because it's between humans and the tiniest organisms.
The howl of a dire wolf.
Dorset lamb may represent a major landmark.
I did not start off planning to design proteins.
Scientific breakthrough will probably be something that we hard to imagine.
Test at birth could map out an individual's entire future risk of disease.
So on.
Got it. Take us back to like where all this started, right? I mean, you're kind of there at almost the beginning, or like the very beginning of the whole creation of the synthetic biology field.
Yeah.
What was it like in the early days?
It was strange. It was fun. We were trying to figure out what it is exactly. So the way I got into synthetic biology is that I was working on this project called amorphous computing. And so amorphous computing was headed by Jerry Sussman, Hal Abelson, and also Tom Knight. Mm-hmm. So this is halfway through my PhD. And the idea about amorphous computing was how would we, and this was it was a computer science project rather than a biology project. How would we program lots and lots of elements that would be small, have minimal power, minimal computational capabilities, but could communicate with one another locally? How could we program environments like that where you have massive amounts of small computing elements? Mm-hmm. You can maybe embed them in dust, smart paint, smart dust, those kinds of potential applications. And as part of the amorphous computing project, I was looking for ways to be inspired. What is the right programming set of abstractions? What's a programming paradigm? And so I looked at biology as a way to be inspired on how to program amorphous computing. Mm-hmm. And so I was doing all kinds of simulations of biological processes like developmental biology, formation of neural tubes, somite formations, all kinds of things in really early embryogenesis. Mm-hmm. And one day I said to myself, I was actually walking down the street, what I want to do is actually flip the arrow. Mm-hmm. And so rather than take what we can, what we know about biology or what we seem to know about biology and program computers, I want to flip the arrow and take what we know about computing and actually try to program biology. And so Tom Knight was interested in doing that. He had a really cool paper with Jerry Sussman on how to build digital logic devices using gene regulation. Mm-hmm. And so I teamed up more closely with Tom Knight and helped him set up the wet lab in, actually it was the AI lab at MIT.
Interesting.
Okay, so it was, Tom Knight was a PI and I was helping him. We had a couple of other people there. And we were computer scientists. And we had no clues what to do and how to program biology, how to actually even work really with biology. Mm. And so we basically figured things out almost independently. So the main big question was, or the skill that we needed was, how do you build plasmids? How do you build DNA plasmids, right? Mm-hmm. And so we actually that was kind of the main hurdle in the beginning. And I was actually trying to build my first plasmid, just not having success, and basically working like 24/7 for 6 months. And I was like, oh man, I made a big mistake. Shouldn't have tried to do biology. Mm. And then we were talking to people and finally said, we got to have somebody come and show me how to actually build plasmids. And so we hired someone. So we knew someone's daughter. She was a graduate student at Scripps. So we hired her to come. And 2 days before she came, I actually managed to build my first plasmid. And so that was kind of the beginning of my journey into synthetic biology. And there were at least 2 other labs kind of interested at the same time. So Jim Collins was beginning to be interested in synthetic biology. So he had a very important publication on the toggle switch, and Michael Elowitz was also working on it with Stan Leibler, and he had the oscillator. And so, that was really kind of the beginning of articulating the notion that we want to build circuits, genetic circuits, that do non-trivial things in biological systems. And first time I talked about it, I talked in front of a computer science audience, and I said, I want to build logic circuits, and here's how I build a NOT gate, and here's how I build an AND gate, and here's how I build like a memory switch. I gave it in front of a computer science audience, and they were like, yeah, sure, that sounds good. And then I remember my next talk, I gave it in front of a biology audience, and they looked at me like I came from outer space. It was like, why do you want to do that? Why would you want to turn bacteria fluorescence on and off? Like, this kind of makes no sense, has no value. And so that was from different fields. We were getting completely different responses. I think the issue was that biologists didn't really see why we would want to be able to build these kinds of circuits. Like, why would you want to make bacteria that blink? It wasn't really obvious. But I think over time, we were able to articulate more precisely why circuits actually matter. What can you do with circuits that you can't do otherwise? And I think once we were able to connect that and be able to articulate, for this field, like for cancer, for development, for vaccines, for autoimmune diseases, here's circuits that can actually make a difference. I think once we were able to do that, all of a sudden, the connection began to make sense. And biologists say, oh yeah, I could use that, or I could see how I can modify that to use that for my own purpose. So I think making that from an abstract, let's build circuits, to a concrete, let's build a circuit that does this for this indication or for that indication, I think that's when it began to really make strides and achieve a much wider audience.
Was this always the intention, or did you, were you more inspired by just scientific curiosity of, how can we develop our capabilities, and then later had to sort of sell this and package it and find value for society? Or was there always this long-term vision around, we can make therapeutics out of this, for example?
Yeah, so for me, even going back as a young kid, I always was a programming nerd. So I just loved to program. And so the obvious thing was to program computers, right? So my first interaction was to program mainframes with punch cards. So that's, I think I was 7 or 8 at the time. So I would go and, create these, I don't know, have you ever seen punch cards? Have you heard what that is?
I do.
You have any idea what that means?
I remember there were these punch cards to collect blood from.
Yeah. So these would be envelope-size pieces of paper, like cardboard. So it was reasonably solid, and on each envelope, you would basically punch in holes, and that would indicate a specific line of a program. So it would actually encode a specific piece of code.
Okay.
And so each envelope size cardboard encoded a piece of code. And what you do is then you created a stack of them. And so the stack of these envelope sized cardboards would represent your program. And you'd walk around with a stack, and then you'd feed it to this machine, a mainframe computer, and it would kind of go through these, make a really cool noise as it was reading them one by one, and that's how you program the computer.
Wow.
And so you fed it this code, it would store that in its memory, and then it would execute your program. So that's how I started programming.
Wow.
Okay.
I think it's also interesting that you were just generally like a computer scientist before you got into biology.
Yeah.
Nowadays, there's a lot of examples of people who do this because the connection is more apparent.
Yeah.
But at your time when you started, it wasn't. These were very disparate fields.
Yeah.
I think it's interesting. But then now, sort of, you're designing these gene circuits. Biology's progressed a lot. Like plasmid design is relatively easy. Like it's something that like RAs do. Like, what were the steps along that path? Like, why did it become easy? Why is the connection between computation and biology becoming more apparent nowadays?
So it's become easier for multiple reasons, and on different levels. Okay, so we can kind of start from the more like experimental level, the production level aspect of it. So back when I was building my first plasmids, there wasn't really a simple way to create new plasmids. Okay, so you basically it was very ad hoc. So you take a piece of DNA, and then you'd have to find enzymes, restriction enzymes, that would cut the DNA in specific places, and then you paste in a new piece of DNA that fit right there. And so there's a lot of ad hoc cut and paste DNA operations. And it was hard. And every new piece of DNA that you wanted to make was a laborious, and then you made that one and then you realize, oh man, I made a mistake because I made this one and now I can't make that new one. So it was it wasn't like a process that was suitable for scaling. It wasn't a process suitable for sharing. It was like everyone was like felt like a one-off. And my story about this, how it began to get a little easier, is that I went through all this pain as a graduate student, and Tom Knight, who was my PI, saw all the pain that I was going through, designing so many plasmids, and every time like really struggling with each one of them. And so he actually came up with this notion of biobricks. So biobricks were now a way to scale and kind of formalize and make the process of building DNA really one that's where it's standardized. So that you make a biobrick, and that piece of DNA can then serve as another to make another biobrick, another biobrick, another biobrick. And so the whole process of making complex genetic circuits became much more accessible. Biobricks, and so I think that was a huge transition. So now there's a standard way to create bigger and bigger pieces of DNA, and after that there were new technologies that improved upon that. So like Moclo and Gibson assemblies, and so technologies that make this thing very powerful and also very convenient to do. And so now when people start building DNA, say, okay, just do Golden Gate, do Moclo, there you go. And this is something you can learn in 3 days. And you can be really good at it, and you can make huge pieces of DNA. Okay? And so just that process from ad hoc to standardized DNA assembly has made it easier for people to, and that's why anyone can come into the lab. I teach undergrads how to do this. People never held a pipette before, and the first time they do it, they're already getting really high efficiency DNA assembly. So that is a critical foundation, experimentally. That's how you can make plasmid. Now, what you make is also really important. So, and that has now become the difficult part. So it's not can you make it, but if you make it, does the DNA do what you want it to do? And now all of a sudden you have to know biology. You have to have a sense of, if I take this building block, this genetic building block, and combine it with this genetic building block, and I put it into this kind of cell, what is it going to do? What's going to happen when I take two of them, put them together, 3, 5, 10? And so now that's the difficult part, is understanding what it is you want to build, being able to predict what will work, what will not work, or coming up with libraries and say, I don't know exactly what's going to work, but I'm going to build 1,000 variants. I'm going to build 1,000,000 variants or 1 billion variants.
Yep.
And so what you have to do is think about, okay, let me try to get something close enough, maybe develop a library. And you have to think about ways where the biology can help you find the solutions, the things that are actually going to work. So there's all kinds of really interesting things that you can do in biology that are not necessarily doable with computers or other kinds of engineering disciplines.
Mm-hmm. I think this kind of brings us to the concept that there's a lot of computation happening in cells already that we don't really consider as computation, and yet they do some phenomenal things. Like we are just constant cellular computation happening at scale. And therefore, if we can better understand how to manipulate this computation, we can also prevent a lot of disease, also eliminate cancer. But I want to sort of concretize what gene circuit design can mean for our audience and just the wider human population. Like, what are the applications of this? How can we manipulate our biology to sort of be resistant to disease or sort of to create therapeutics? Things of that nature.
Yeah. So it's a great, compound topic. I think the first aspect is that regarding what happens in biology as computation, I think is really empowering. It really changes what you perceive as actually taking place and how you can approach both understanding it, but then also manipulating it, modifying it, getting it to do what you want. It's also the fact that the computation that's taking place there is not for the sake of computing, it is actually doing the thing it's supposed to be doing, right? So whether it's regulating genes or regulating metabolism. So computation and metabolism, for example, in a cell, are not distinct. They're really interconnected, right? They're the same thing. Or assembly of structures, biological structures, lipid bilayers, cellular membranes. Compartments so the physical processes and the computation are completely integrated together. Sometimes they're one and the same. Whereas in computing, you regard the computation as this kind of separate modular thing, and then that interfaces with your mouse, your keyboard for input, that interfaces with your monitor for output, that interfaces with many different things, but it's kind of a very well-defined interface. Whereas that interface doesn't exist in biology. Everything is connected. Everything is doing all things, and so that's why you have to treat it slightly differently. You have to think about it slightly differently. But the concept of biological systems are computing systems with controllers, with inputs and outputs, with sensors and actuators, with logical elements that make decisions. I think once you get comfortable with that notion, and that notion has been around before synthetic biology. I think synthetic biology has now taken that notion and said, okay, these are the existing things that biology already does. Let's modify them, right? And so it enables so many capabilities that otherwise wouldn't be possible, I think, without synthetic biology. So there are situations in biological system, something goes wrong. It could be one base mutation somewhere. And so if you want to fix that, great. So you can come in and maybe use CRISPR or prime editing or something like that, and you modify that specific base or a few bases, and you can treat a disease. But many, many other situations in biology are much more complex than that. They're not just a single element that went wrong. It's really failure of a system, series of many mutations. And obvious one is cancer, where sometimes it's a single mutation, but oftentimes it's a large set of mutations, and they could be different in different people. And one patient's cancer mutation could be very different than another patient's mutations. And so it becomes a system level problem. It becomes a situation where you can't find, oftentimes you can't find the magic protein or the magic chemical that will fix the problem. And so the system level approach for cancer creates a situation where you can say, let me understand that it's a system level issue. And now let me design circuits that can address it at the system level. For example, say that you want to be able to create circuits that can distinguish cancer versus non-cancer cells. And oftentimes you can't find that one distinguishing biomarker. You really have to think about what's the collection of biomarkers and sensors that I have to build. And you say, okay, once you think about it as a collection, I can say, oh, there's a collection 2, 3, 5, 6, biomarkers. And if I can sense those biomarkers and can determine the levels of those biomarkers, I can say for sure with 99.99% confidence that's this kind of cancer cell, and then act accordingly. Kill that cell, but don't harm healthy cells. Right. So I think that's one example, and there's several other examples with similar nature. Right. That when you think about this is a system. Something is going wrong with the system. Let's figure out what has gone wrong by having a complex system that can detect. That's your synthetic biology system that can detect what went wrong with the biological system. It's system to system, right? You want the system to understand what the other system is doing, right?
I see. Yes.
That's really the key.
Fascinating.
So you want to program biology with things that are as sophisticated as the biology itself.
Understood.
Right.
Okay.
Right.
Yes. And I think this is particularly a challenge where you don't have this single mutation that's driving the sort of cancer tumorigenesis, but rather you have either, yes, as you said, like a collection of mutations, or there's just a dysregulation in the actual levels of different genes being produced.
Right.
And let's say these are cell surface receptors that you can actually sense. The problem is that a normal healthy cell, a self cell, will also express that same receptor to a certain degree. It's just that the cancer cells express them much more vigorously at a higher density. And so an example of this, I imagine, would be trying to sense the density of this receptor versus this other receptor in order to distinguish, yes or no, is this a cancer or not? And this kind of, if you just scale this higher and higher, it resembles a neural network, right? You're creating a neural network that performs computation, then has inputs, it calculates, and then it makes a decision either way, right? Is that an accurate description?
Yeah. Yes. Let's further explore the cancer example. I think that's a great example to begin to think about what synthetic biology, how synthetic biology approaches specific problems. So when we started working with cancer therapy, we said, okay, what we're going to do is we're going to build logic circuits that can identify cancer versus non-cancer cells. So we're going to find biomarkers and say, if this biomarker is high and this other biomarker is high and this one is low and this one is low and one is low, then it's a cancer cell, right? And so that actually looks at the biological cell as binary almost, right? So that's how we started. And we said, okay, low, kind of working on lows and high signals. And in certain cases, that's perfectly reasonable. In certain cases, you can say, I can uniquely identify this type of cancer by treating things as binary, by saying these biomarkers have to be high, these biomarkers have to be low, and if that's the case, I know with almost complete certainty that that's a cancer cell type. But in many other cases, the cells are not binary. They don't operate just as high and lows. They operate really with a lot of intermediate values. They have these complex networks that regulate things at intermediate graded levels with noise and probabilistic features. And so what happens is that when you realize that, you say, well, these kind of binary circuits, digital logic circuits work fine in certain cases, but are completely inappropriate for detection of these other types of cancers, these situations where things are not quite as clean. And so we've been, this is something we've known for some time and appreciated for some time, but we didn't quite have a solution. We didn't have a mechanism that would reliably look at these complex situations and say, okay, I can engineer new kinds of circuitry that will be able to work in these analog, fuzzy, noisy environments. And a few years ago, we realized that we can actually create genetic circuits that operate not as binary digital logic, but rather emulate artificial neural networks. So the notion that rather than building AND gates, NOT gates, OR gates, and so on, we can actually build genetic circuits that operate using the building blocks of artificial neural networks, which are things called perceptrons. So we figured out, and these perceptrons work using analog values. They do analog computation, analog control. And we realized, we know how to make perceptrons inside living cells, and we actually know how to connect perceptrons inside living cells. And all of a sudden we have the ability to create inside the cell artificial neural networks. Okay. And so we're kind of inspired by, AI and so on. We said this is, because it's such a powerful paradigm, let's actually try to use that paradigm to program cells rather than the dominant paradigm, which is logic, digital logic circuits. So as we began to build them, we also began to identify kind of similarities with the way biology actually works. So it wasn't necessarily that we said, we're going to build artificial neural networks because that's the way biology works. We said these are powerful networks. We want to build them. But as we built them more and more, we looked at them side by side and we said, hey, the artificial neural networks that we're building, they look like biological networks.
Exactly. Yeah.
Right? They all of a sudden it seemed like, we're actually talking the same language. What we're building uses metaphors, system wiring, the way you connect things looks very similar to what you find in papers, in textbooks that describe normal biological networks. We're like, oh my God, this is the same language, right? And so that, that was a really cool kind of eureka moment for us. And then we began to realize that because we are speaking the same language as biology, that all of a sudden changes how easily we can understand what's going on in the biological system, detect it, and control it. So the interface between digital logic and biology has always been a bit of a struggle, right? A bit incompatible. A bit incompatible because you're forcing biology to sometimes speak, to do things it's not really great at doing. Whereas if you look at the artificial neural networks that we're building, and then you put them in a cell and you connect them to biology, it's almost like a seamless integration. Right? And they're speaking the same language. That means a conversation between your synthetic circuit and the underlying biology becomes much better, becomes much cleaner, becomes something that makes much more sense. And so that really has been transformative to the way we think about synthetic biology.
And it makes sense in retrospect, right? Like the reason that biology is perceived as such a complex field, and it's sort of like a black box that we're just investigating at one level at a time, one relationship at a time, is just the fact that it's a neural network that's just ineffably complex. And maybe humans just aren't built to understand the big picture in the way that a perceptron or a system of perceptrons can, right? Talk a little bit more about that if you have any other points, but also just the distinction between an analog versus a digital circuit. I think that would be
So digital systems, binary systems. So they're actually built on analog levels, substrate. They're built typically implemented with electronics and let's say with voltages. And so voltages are not just low and high. They have analog values right? You can have 0 volts, you can have 1.7, you can have 2.3, 4.2. You can have the whole analog range. But what's been transformative in terms of moving from that analog to digital, so computing has actually made the move from analog values to binary low and high values, and that's been very powerful. And so what happens is that you say, okay, I'm building systems that work on analog values, because that's the physics, right, of electronics, right? But you say, if the values are between 0 volts and 0.7 volts, I'm going to regard that as low. And if it's 3.5 to 5 volts, I'm going to regard that as high. If it's somewhere in between, that means it's a forbidden zone, not allowed to be there. And so once you begin to associate values, zeros and ones, lows and highs, to these analog levels, and you can say, I'm going to then build input-output functions that transform, that take input values and then make output values. For example, let's say an AND gate would be one that a 2-input AND gate would be one where you have 2 inputs coming in, 1 output. And so it looks at the values of the inputs, and if the value of the inputs is, let's say, above 3.5 for both inputs in terms of voltages, then the output will be 5 volts, right? So that becomes a 2-input AND gate. If my 2 inputs are high, then my output is high. Otherwise, my output will be low. Okay, so that's basically how you create a digital logic AND gate. Now, it's powerful because you can take one of those, and as a human, you can say, I'm going to take one of those, I'm going to connect it to another one. I'm going to connect it to another one. And every time you make the connection, the system still works on zeros and ones, highs and lows. So it's easy for you as a human to think about, I'm going to program this very complex behavior, and I'm going to be able to map it to zeros and ones. It just, it just makes sense. It's easier to keep track of. And that has allowed the creation of these incredibly powerful computational systems because it's composable, it's predictable, it's scalable. So I can build a laptop, that has billions of computing elements, and it becomes very reliable, right? Until you begin to run, I don't know, Microsoft Windows or something, right? But it's something really reliable. And so that has enabled the information revolution. Now, biology was not designed in the same way. Right? Biology was not designed to work on zeros and ones. Right? Biology works under different constraints. So biology actually works on constraint that it doesn't have the luxury of connecting 1 billion elements together and operating like that. It has to be super efficient. It has to utilize every resource that it has to its maximal potential, right? And the way to do that is actually to build analog systems that operate at intermediate levels, not just high and low, because that's wasteful, right? You need the intermediate values. So that's one thing. Right? So to make more optimal systems, you need to use intermediate values. What else do you need to do? You need to make networks that control things, make decisions, but you can't just say, okay, this protein is only going to be used for developing a liver cell. Almost everything in biology is multi-purposed. So that same protein will do one thing, but in a different context, it's going to do something else. So it could be that protein is used to develop liver, but at the same time, that protein could also be really important for helping immune cells do something completely different. So it's very contextual. There's a lot of reuse of the elements in many different contexts. That's much more efficient, right? It's much more efficient. And so that's why evolution has resulted in systems that are analog and are complex and highly interconnected and where their parts are used for many different things. And so the evolutionary process and the need for optimization has resulted in that networks that are complex, highly optimal, and very difficult to understand, right? Very much like artificial neural networks.
Exactly.
Right?
Yeah. Very hard to dissect.
Yeah.
But I think this is very similar also to the analogy that the intelligence of these like large language models is like roughly proxied by how much they can compress information into as few nodes as possible, right? That's like the hallmark of intelligence.
Right.
I think to understand how this works at like a basic level, like we've talked about AND or circuits, but how does one go about making the first AND circuit, the first OR circuit at a biological level, and then how does this grow over time to become something that's more analog and sort of programmable still by humans? Yeah. So when I started, as part of my PhD, was to define the basic building blocks for creating genetic circuits. And so what I started with was the most basic yet non-trivial digital logic building block, which is the NOT gate. So I think this is a good place to begin to think about how do you actually build genetic circuits. And so to build a NOT gate, in principle, I would call it quote unquote easy, where you basically have one input, one output, and the output is the inverse of the input. What does that mean? So the input can be either high or low, meaning there's a high concentration. We're going to use proteins to represent these concentrations of the inputs and output. So you have a protein that's on the input. It can be low or it can be high. And then the output has to be the reverse of that. And so, as part of my PhD, I said, well, this is with Tom Knight, I had said, let's take repressors. And these are proteins in the cell that basically repress the production of whatever you connect them to. And so if you have a high level of the repressor, it represses the output. So you have a low level of the output. And if you have a low level of the repressor, then it doesn't repress the output. So there's a high level of the output. And so by having a repressor and connecting it to an output, you can begin to realize the basic building block of digital logic circuits, the NOT gate. Okay, so that's the basic block. And so then you can take a NOT gate and you can actually connect it to other NOT gates. And so there are ways, so for example, to build an AND gate, you can actually do that with a few NOT gates. Okay. Interesting. How does that work? Maybe it might be too complex to talk about.
Let's have fun. Let's have some fun and go through that. Okay. So visuals help here, but let's try to do without the visuals. So let's think about it. We have the NOT gate. It works. We have a repressor, and then we can have a repressor repress the output. So that's the basic building block. And the cool thing here is that now you can build anything that you want out of that. So let's think about that. So a 2-input AND gate is one where if both inputs are high, then the output is high. Otherwise, the output is low. Okay. All right. So we have that. So we need 2 inputs. So we're now going to use 2 repressors, right? So rather than a 1-input NOT gate, we're going to have a 2-input AND gate. So we have 2 repressors. They're both inputs. And what we do is we connect it so these 2 repressors each one of them represses another repressor. So now we're bringing in another repressor to the mix. So we have repressor 1, R1, R2, and they both repress R3. Okay? And then R3 represses the final output.
Okay. I see.
So the way it works is that if both repressors on the input are absent, then you have the intermediate repressor unrepressed, is high. That means it represses the output. Okay. If one of the repressors on the input is absent, but the other one is present, you still have production of that intermediate repressor. You're still repressing the output. The only time where the output is high is when both input repressors are high. They're both repressing their own copy of the intermediate repressor, There's no intermediate repressor. That means there's a high level of the output. That's a 2-input AND gate.
I love that. Fascinating.
Okay? And then once you have these AND gate, 2-input AND gates, once you have a NOT gate, it's universal, meaning that you can, in theory, build any kind of digital logic that you want.
Amazing.
And that's, once you realize, you're like, oh my God, this is so cool. At least I thought so, and a lot of other people I talked to as well. So once you realize, you're like, I got to program cells. I have to be able, I know, okay, I see how to program them. I can build circuits out of that. I want to program them to do X, Y, and Z. And so that was, so that's kind of the digital logic aspect of it.
How far have we taken the digital side?
Yeah.
Like, what's the most complex digital circuit that we've created in biology?
So, so the problem with digital logic in biology is that you run into kind of scalability issues. It actually begins to become difficult to build more and more complex circuits. I'm not saying it's impossible, but it certainly becomes more difficult. So some of the bigger circuits, so for example, Chris Voigt, Tae-Sak Moon, postdoc in his lab some time ago, created a 4-input AND gate, in bacteria. So that was, at the time, that was the biggest circuit. So I would say, in terms of the digital logic aspect, it's been, there was really nice progress. I actually was tracking the size of the circuits that were built at some point. And so at that time, Chris Voigt's was the biggest one. We had some previous ones that were the biggest at the time. But then it was interesting that there hasn't been as much progress. There wasn't like a Moore's law in digital logic circuit sizes. So some people said, okay, once you get 2, then you can get 4, then you can get 8, 16. We're going to have a Moore's law, like exponential increase in the number of circuits that you can, the number of building blocks, digital logic gates you can have in cellular circuits. Wasn't the case. And it really kind of plateaued, and people haven't been pushing them further and further up.
Is the reason for this, like from an intuitive perspective, just that like digital electrical logic gates and circuits and transistors like shielded from the outer world, right? But all of these systems in biology are noisy.
Yeah.
You're essentially anything you put into a biological system is floating in this like molecular soup and sort of being impacted and by the other molecules around it. Is it noise that just amplifies as the circuit grows?
Yeah, a great question. So there are multiple reasons, I believe, that explain why it hasn't been that easy to keep on going up and up and up. One of them has to do with just availability of good parts. So it's actually not that easy to create more and more really good parts that are really good and on and off, right? And they have to be orthogonal, meaning that you want to make sure that they don't cross-repress, that this part only acts in a modular way. And so finding large sets of orthogonal parts that was and still remains a challenge to an extent. Cellular resources. So whenever you build more and more digital logic circuits in a computer, they consume more power. But you plug it into the wall, you have lots of power, right? So power is not an issue. But whenever you're doing it in a cell, then many cells, especially bacterial cells, are running almost at full capacity. They don't have a lot of power to give. But then you're asking them for more and more power, and they begin to resist. They begin to push back, right? And so they don't like it. And so they don't have excess power to give to you, and so they might begin mutating your DNA that you put in there. And so the power consumption becomes an issue as well. One of the things about these digital logic circuits is they need a lot of power from the cell. So to be really good and on and off, right, you need the off to be really low, but you need the on to be really high, because you want to make sure the on remains on, right? So even if it fluctuates, everything fluctuates. So the level is going to fluctuate. So we want to make sure that the fluctuations don't go too low, and so to make sure that never happens, you push it really high. So you keep the levels very high in terms of the levels of the protein. When you keep the levels high, that means you're producing a lot of proteins. That's a lot of power that's required for that. And so digital logic circuits require a lot of power. Cells don't love that. And they start, like I said, they start pushing back on that. And that's been one other reasons why it's been kind of hard to create digital logic circuits that are larger. And also it's a DNA, by itself, sometimes hard to deliver. So multiple reasons, but these are some of the main reasons. So one of the things we're excited about is that these analog artificial neural networks, they don't have to operate up here. They don't have to, each one of them, they don't have to operate maximal high levels, high power. They operate like the cell does at intermediate levels.
Okay.
They're good if it's, low intermediate, slightly higher than intermediate. And so I'm hoping, it remains to be seen, that by programming it, the levels, the power levels that the cells normally operate, we might actually be able to create larger and larger circuits and being able to really scale up our artificial neural networks. I'm not saying forever. We're going to, there's also resource issues there as well, but maybe a little more. Maybe get them to be a bit more complex.
Fascinating. What's the work that we've done on this so far? Like you mentioned that it's this is a hope.
Yeah.
But have we started on this? Is there any initial data that looks promising?
Yeah, so we've been able to build small artificial neural networks. So we've been able to build a small library of basic building blocks of perceptrons, and then connect them to create 2-node and 3-node circuits, networks inside single cells. So right now the biggest one that we have is still a 2-layer network that has 2 components on the first one and 1 component on the second one.
Okay.
Right, and so that's where we are right now. We'll see. Again, I can't predict how far we can push it. Yeah. Right, but we're hoping to find out more about that. As we build more and more. So right now we just are introducing the 4th element and making sure that's completely orthogonal to the other elements, and then use that. So build another one and another one and another one. But interestingly, even with 3 nodes, 3 perceptrons, because they have so much customizable features to them, we can already get a huge number of different behaviors, even just from 3 nodes, and really customize and fine-tune them to create the exact kind of input-output behavior, analog input-output behavior that we want.
That's amazing. It's truly the beginning of the field. I mean, I think I hope one day we have biological neural networks within cells that are as large as GPT-5.
Yeah.
But we'll have to see. I'm curious, from a biological concrete perspective, what is the actual difference in the construction of a perceptron as opposed to an AND gate or an OR gate?
Yeah. So what you need to do in order to build perceptrons is first think about what is the computation that is performed by a single perceptron, as opposed to a digital logic gate. And interestingly, actually, perceptrons conceptually are not that complicated. So a perceptron, what does it have to do? It has to be it has inputs, right? And the inputs have analog values. And so it has to first be able to add input values. So you have an input x1 and you have an input x2. One thing that you need to be able to do is add x1 plus x2, an analog addition. Okay? So actually that's not that hard to do in biology, because what you can do is basically create it so the x1 and x2 make the same RNA, for example. Okay? And if they make the same RNA, and as long as you're within the operational range, if they make, it's an addition. Okay? So addition, fairly easy. You need to be able to do subtraction. That one was a little more difficult. So the way you do subtraction is you have one RNA molecule, and then the other one that's doing subtraction, you make a protein that binds that RNA molecule and destroys it. But one at a time. So one protein destroys one RNA, and that's it. And it remains bound to it. So that effectively gives you subtraction. Right? So you have one that's the positive, and the negative one just comes in and essentially sequesters that. It comes in, binds that RNA, that protein binds the RNA, and moves it out of circulation.
So you use like an siRNA or?
So we're using these things called endoribonucleases.
Okay.
So these are RNA binding proteins. Actually, they're CRISPR, from the CRISPR family. But they don't do any DNA editing. They don't touch the DNA. They only bind to RNA. And the cool thing about them is you can say, here's an endoribonuclease, and I'm going to put an endoribonuclease target site on a piece of RNA. It recognizes it, binds it, destroys the RNA, remains bound to it.
Okay.
So it's doing a subtraction operation.
And did you have to engineer this to remain bound? Because usually they keep cutting.
Yeah, so that's really important, is they don't act like enzymes that keep cutting, but they do, it's effectively a single turnover enzyme. Only does it once.
Okay.
So it comes in, and so we did not engineer that biology gave that to us. Okay. And so now we have addition, we have subtraction using biologically available components. So we have addition, subtraction. We can also have weights on them. So we can have addition that has a higher weight, addition has lower weight, so subtraction with weights. And that's by modifying various things on the RNA, making more protein, less protein, more RNA, less RNA. So that's addition, subtraction, weights, are necessary for a perceptron.
Yeah.
And then you also have to have this, what's called an activation function that is nonlinear. Okay, so that one sounds complicated, but it's actually not complicated. Nonlinear activation function. So specifically, again, complicated name called a rectified linear unit, simple math. Okay, so basically, imagine you have x2 minus x1. So we're doing subtraction. And so if x2 is greater than x1, fine, that's the value. Right? Those are two inputs. But if x1 is greater than x2, then the mathematical value is less than 0, but we can't get less than 0 in biology, so it becomes a 0. And so what happens is it's a nonlinear function, because if x2 minus 1 is negative, the output is really 0. And if x2 minus 1 is positive, it's just that value. So it's nonlinear. It's flat, then just diagonal. And that's really important for artificial neural networks. So again, going over it, addition, subtraction, weights, nonlinear activation function, that's a perceptron. And the other thing you need to do is be able to connect it to other perceptrons, and you have artificial neural networks.
Amazing. Yeah.
And so and it's basically the same kind of thing over and over and over and over and over again, right? And it's all about the connections between these. So it's basically a perceptron network. Once you can create a perceptron network, you have an artificial neural network, which is nice. The power of artificial neural networks is the fact that you can modify the weights. And by modifying the weights, all of a sudden it can do something completely new, right?
Yes.
And so, one example might be artificial neural network that can detect cats, online, like in pictures. Okay. And then slightly modify the weights, and maybe it can detect dogs or cows or boats or cars, right? And it's all about, you have a complex network. It's about modifying the weights to make it do something. That's basically learning. That's what learning is all about. Now, if you're to try to build a computer program that was able to detect between, cats and cars or whatever, right? Could you write a program like that?
Very difficult.
Very difficult.
Yeah.
Very difficult.
Or at least not generalizable. Yeah.
It's, how would you do it? It's just, if you think about all the possible ways that cats can look versus cars, just was a one letter difference, but you couldn't do it, right? From those pictures. The beauty of artificial neural networks and why AI is so powerful is that you can just give it a whole bunch of examples of cats and cars and other things, and it learns. And then it figures out the weights, and look at that. You can do something that us as humans, in a rational way, either would have a really hard time or would not be able to do that at all. And that's been the thing that's been transformative about AI, is that you don't have to understand the solution. You just have to give it enough information, have it change the weights until it figures out an algorithm, which then can give you the solutions incredibly reliably with incredible precision. We don't know why. We don't know why that exact set of weights creates a good cat classifier, a good car classifier, right? But it works. And so what we're saying is that's awesome for computing. Let's bring it back to biology, where it really belongs, right? So, AI was obviously, inspired by biology. And it's been completely transformative for computing and our existence, I would argue, or will be, right? Or in some cases already is, obviously. But I think that having that capability be brought back into the biological discipline, and now being able to train biological networks to be controllers for biological networks, right? So synthetic biological networks that have been trained because they're artificial neural network to work with these incredibly complex environments that we only slightly understand. That's why, as I mentioned before, now they're going to be speaking the same language. They're going to be trained so that this one, the synthetic one, can be trained to work with the native one, the endogenous biological network, right? The natural one, right?
Got it. This is fascinating and definitely the future, I would say. Very much have been increasingly swayed in the direction of synthetic biology and circuits for a while now. I'm curious, though, there seems to be a few differences and barriers to overcome. Like one being that, like unlike in an artificial neural network that's in a computer, each of these perceptrons needs to operate using like non-overlapping molecular machinery. Otherwise, they're in the same molecular soup, but they're going to interact. But like presumably, if you make enough of these perceptron variations and interchangeable sort of input sites that you can easily modify, then you can stack these on top of each other, and hopefully the fact that they're graded allows you to condense a lot more of them into a single cell than what you were able to do with sort of digital circuit design. Firstly, does that make sense? Like, do you have a strategy on that? And then, like, the last question would be, how do you do gradient descent in this system?
So the first one, I'll have an answer for. The second one, not yet. Okay. I mean, we have ideas, but not yet. So with respect to the first one, right now we are certainly trying to create a set of orthogonal components. As you were mentioning, ones that would work well in a somewhat modular fashion, analog, but somewhat modular fashion, right? So whatever this one does, whatever this perceptron does, is confined and doesn't affect the random perceptron, only the perceptron that's supposed to be connected to, right? So well-defined connections and interfaces and kind of modularity. So we're trying to certainly create elements that are modular, and it's doable. I mean, biology will support modularity, at least to some extent. Not there's not full modularity, but at least partial modularity. Now, this is the way we're doing it now, but I do believe that it doesn't have to be that way. So if you have a bunch of components that may have some crosstalk between them, fine. So just train your artificial neural networks so that they can deal with that crosstalk, so that they can use that crosstalk. And guess what? My belief is that if you train them properly, they'll figure out how to either not be harmed by it or even utilize. Biologists figure out how to do it. I would argue the artificial neural networks should be able to use that as well. So I think that as a constraint is only a temporary constraint, and we will be able to actually create very powerful systems out of components that may have interactions between them, right? So I definitely think that. Now, the circuits that we're building now are designed by the computer. So we basically have biocompilers that where we have them learn about the parts, and we have them learn about how parts come together, and then the biocompiler says, we tell the biocompiler, we want something that looks like this, that has this, can recognize this shape. It can recognize this shape, and the biocompiler can now say, okay, fine, here's a DNA where it implements an artificial neural network that can recognize that shape. So we now have the ability to do that. That's actually as of a few days ago.
Fascinating.
So we've been able to actually, give it shapes that we've never had before, that we've never designed before. Actually, some of them we weren't even sure that we could make. And then the biocompiler said, well, try this. And so we're now, testing that out. So that process still happens in silico. But ultimately, we definitely want the cells to learn. Right? We want that learning. Right now, the learning and the design are happening in the computer, then used to build DNA. We ultimately want that loop to be a closed loop within the biology, so that the cells can actually modify their own DNA and ask the question, am I doing better? Am I doing worse? Gradient descent, right? Type of style.
Crazy.
So we now have initial ability to actually have cells that can modify their own artificial neural network weights. And so now they're able to actually modify what they're doing. And we have some ideas about how they can evaluate whether they're doing better or not. So towards this goal of cells that learn and do a better job, we're going to get there.
I hope so.
Yeah.
I don't doubt it. Fascinating, amazing. I think more people should be in this field. It's still somewhat under-researched, I think, underrated as a field. We don't have a lot of time before you have to go. So I wanted to touch on two other applications. One being that your lab is quite an entrepreneurial place. And I think you have this intention of having it continue to be an entrepreneurial place, to be and come be a student or a postdoc. And one very successful example, sort of Strand Therapeutics, recently raised 150 million, pretty good news. So congratulations on that. You co-founded this company with Jacob, who's also coming on the show actually, which will be fun. But yeah, how did that happen? Talk us a little bit about the sort of application of this self-replicating mRNA sort of in this sort of cancer field. And then we'll also talk briefly about the React project as well.
Yeah, excellent. So with respect to cancer, from the very beginning, it seemed very natural that cancer would be an application for synthetic biology. So I think once once I realized we can program cells, one of the first things I said, I want to be able to program circuits that can detect cancer versus non-cancer cells. Had no idea how to do it. No idea, but I was like, okay, this seems like this makes sense. And so that's an area we've been interested in, my lab's been interested in, from the very beginning, but we had to spend a long time on building the foundation to be able to do that. And so we did that. We actually had a project when I was had a faculty position in Princeton, our iGEM team. Hopefully folks know what the iGEM is, the International Genetically Engineered Machine Competition. So we had a team on undergrads, and a project that we came up with, I think it was back in 2007, was let's build cancer cell classifiers, circuits that can detect between cancer and non-cancer cells. I also had a very early collaboration with Kobe Benenson. And so we worked together and we published together on cancer cell classifiers. So we had folks in the lab, Janjit, so various folks working on aspects of that. And so DNA was used as the therapeutic agent. So DNA delivered into the cells. The DNA would encode circuits that can detect cancer versus non-cancer. 2011, we actually pitched this idea to DARPA at the time, and we said, we want to be able to build more DNA based circuits, and the program manager said, why don't you look at RNA, this self-amplifying RNA as an opportunity? And I read about that and I was like, oh my God, this is such a cool delivery vehicle. Why is it cool? So self-amplifying RNA is a piece of RNA that has the ability to go into the cell, and the cell recognizes, it begins to make the initial proteins, and the proteins then that are encoded on the RNA are able to make more copies of that RNA. So you start from a single molecule of RNA, and you can get in that single cell 200,000 copies of the RNA. So all of a sudden you get a tremendous amount of RNA, which is really helpful for several reasons. I can come back to that. The other really cool thing about self-amplifying RNA is that normally a piece of RNA, you get one gene. But the self-amplifying RNA can actually encode what are called multiple subgenomic promoters. And each subgenomic promoter becomes an independent unit of production, of protein production. And so from a single piece of RNA, you can basically encode multiple devices, regulatory devices. And once I realized that, I was like, we got to make circuits on RNA. That's just too cool not to do. Okay. And so we had several people in the lab, Liliana Roblowska, one of them, so Tsuku Kitada is a postdoc in the lab, and he's one of the co-founders of Strand, and Jake Beecraft. And so they created this RNA, self-amplifying RNA platform that is able not to just have RNA and make a lot of the RNA, but also encode computation. Okay? And so now you can think about the RNA as being as powerful as DNA. And we said, okay, if it's as powerful as DNA, well, maybe we can encode some cool things on it, such as cancer cell classifiers. So now the RNA goes into the cell, and it doesn't just produce the therapeutic protein that kills the cancer cell. Okay? It actually performs a computation to ask the question, is this a cancer cell or not? And if it is, then kill the cell. If it's not, just go away. Right? And so the self-amplifying RNA provides you with the power to be able to do that. Okay? And so I've also been working with Daryl Irvine in his lab from the very beginning on this, and he's also one of the co-founders at Strand. And so we made a variety of different circuits on RNA and created different versions of that. And Jake and Tasuku wanted to really translate that into helping real patients. So go from the lab where we're doing experiments, either in cell culture or experiments, in vivo experiments, to really bring it to the clinic. So they had the vision that we'd be able to do that, which is tremendous, and they really made that happen. So they, with Daryl and I, but they really were the main force behind co-founding Strand, and we're able to create this, amazing environment. And just a little over a year ago, they started the first clinical trial with a self-amplifying RNA. So the first in human self-amplifying RNA for cancer, and they were able to report on that. They presented at ASCO, presented really cool, phase 1 clinical data showing that patients that have gone through, all the, latest and greatest immune checkpoints and cancer therapies, where at the end, they really didn't have any more options available. And we are focused mostly on melanoma, and we're able to use the self-amplifying RNA and inject it and have certain cases where the patients had complete recovery, which was amazing. Some of them had partial recovery, and some of them had stable responses. And so that was, just phenomenal to see that. And so there's ongoing clinical, trial on that. And then they're also creating more sophisticated version. That version in particular was injected directly into the tumor lesions. And so now they've reported also on a new version that can do this, more sophisticated computation that would be able to be injected systemically, and it would be delivered to different places in your body, perform the computation, and only activate the cancer immunotherapy in the cancer, in the cancer cells. So
fascinating.
It's just been phenomenal to see what they've been able to do.
Full circle as well from just this let's bring computation to biology question from a long time ago. Very, very briefly, talk to us a little bit about the React project as well, how that's going, and sort of what your hopes are for this as well.
Yeah. So the React program is really fun. It's cool in the sense that it also integrates devices outside, not just strictly programming synthetic biology, but also interfacing with electronics and devices that could be implanted. And so this is a large project that is where Ken Shepard is really leading as a main PI on this. And so the idea with this React program is to be able to both genetically engineer cells and engineer electronics and devices to be able to interact with the cells. And so the idea is using optogenetics, the device, the electronic device, would be able to tell the cells produce a therapeutic or shut it off. It would be able to tell what the cells are doing and be able to actually report that back into, for example, your iPhone. So imagine that you can have this implanted. You have a device. The device can communicate with genetically engineered cells. They will express and produce and secrete a therapeutic molecule of your interest. And you'll be able to have an app on your phone where you can control that.
Yes. I want it.
Yeah, I want it too. So I think it's pretty cool. Just dial in. I want more of this protein, less of that protein. And then it would be able to send, is it doing a good job? Is it more what you want or not? So
I think both of these topics are just fascinating in the sense that, like, I've seen melanoma be removed from patients, for example, before. And it always strikes me as odd that, like, our only tool for this. Just like, let's physically excise it with a glorified knife, right? But to be able to inject something and just have the cancer go away in this sort of non-toxic manner to the rest of your cells, to be able to, if you're like a diabetic patient, for example, rather than having a continuous glucose monitor strapped to your arm or sort of doing a finger prick, literally just having an implantable biosensor that's tracking your glucose levels and maybe hundreds of other things in the long term. And then this like literal collection of synthetic cells that's just separated from the rest of your cells, but it is producing insulin on command with some computation that's either performed sort of internally, given the sort of gene circuit design, or via an iPhone sort of Bluetooth receiver, optogenetics, like so many things from 100 years of biology have come together to like make this possible. And it's like the first example of truly like the collision of digital and biological and basic biology and therapeutics. It's amazing. Ron, last question before you head home. What's a book that you'd recommend to our audience to go deeper or just, I don't know, be inspired?
Okay. I'll go old school on you.
Okay.
To be inspired. Actually, for some reason, I just had a conversation with my son, just reading, and I had as a young kid, I was fascinated with science fiction, and one of the very first books that I read was The Foundation by Isaac Asimov, so and I talked to my son about this book a few times, and just recently he actually read it, and so we talked about it, and so he just loved it. He was like, "Oh my God, this book is just transgenerational and really kind of able to provide a timeless story." And so he loved it. We had a fun time talking about it. I think there's a connection here to biology, or to what we're trying to do in synthetic biology, because it was based on this notion that you can take these very complex systems, right? In this case, I don't remember what it was called, psychohistory, right? And be able to take these incredibly complex systems and be able to make predictions that would be accurate predictions. And so I think there's really fun kind of analogy to what we're trying to do now in our biological systems. Is, can we actually make accurate predictions about what would happen, if we modulate things in systems that are so intricate and difficult to understand. I don't know. So I think it's kind of a timeless book that's worth revisiting and kind of reflect on how that might relate to modern times, if you will.
Or anything from Isaac Asimov. If you're looking for like a shorter version or just something to read as a short story, The Last Question is also pretty good. But Ron, thanks for coming on. It's been a pleasure to have you.
Oh, it's been super fun. I appreciate you inviting me here.
Awesome.